company. So let's talk about here. If you took this recombinant plasmid, so I wanted you to see what the company I ordered our plasmid from actually does. Okay, so remember, you take your DNA from the source of the gene that you want. So in this case, it was human DNA to get the human growth hormone gene. And you cut it out, and then you insert it into the plasmid, and you have to use the same restriction enzymes. Why is it imperative to use the same restriction enzymes to cut both sources of DNA? Why do we have to use the same to cut both sources? Lucas?
Is it so that when you add it back, like the same bases are like following it and like before it, so that it doesn't change?
That's correct. All right, and we call those, that part that's not bonded together, we call those sticky ends, and so they create the same sticky ends and therefore complementary bases, and so they'll stick together. And then to make it permanently stick together, we also isolated an enzyme that naturally is found in nature, and that's DNA ligase. We know that from gluing together what? DNA ligase. What did it put together? The Okazaki fragments. And so then you get a nice intact plasmid. And so then, after you make the recombinant plasmid, the second thing you have to do is to try and get that plasmid into the bacteria. So remember that bacteria has the big circular chromosome, and it has plasmids. Plasmids are found inside there. And then what you have to do is take your recombinant plasmid. So this is foreign DNA to this particular bacteria. The recombinant plasmid in this activity had the human growth hormone gene in it. So what other gene did it talk about that it had in it? Ampicillin resistance. And so the ampicillin resistance is part of the actual bacterial plasmid, but this is the new DNA from the jellyfish, or not, lab is from the jellyfish. In this case here, it was from the human DNA, the human growth hormone. So then, we can't see bacteria, but we can make a clear solution with bacteria in it and put these recombinant plasmids and mix it in the environment. And what will this bacteria do? It will take it in. Bacteria naturally take in foreign DNA. The reason why we use plasmids as a way to get the human gene in is because plasmids come from bacteria. So what happens is when this bacteria takes in this plasmid, it's not going to destroy that plasmid, because remember, bacteria also have naturally restriction enzymes. That's where we get the restriction enzymes to cut the genes out in the first place. So bacteria have naturally these restriction enzymes, but if you use bacterial DNA, it will not cut it up. And so therefore, that's the key to using the plasmids. And so therefore, whatever genes that are on this plasmid, that bacteria now has. And that's one of the ways that bacteria get genetic diversity. Remember that when bacteria reproduce by binary fission, you start out with one cell and it divides into two. Those two cells are genetically identical. So reproduction itself does not provide diversity for bacteria. The original source of diversity for all organisms are what? Mutations. Mutations in the DNA. But then this process, the fact that bacteria can take in foreign DNA and keep it and express the proteins that those genes have or code for, changes the bacteria and therefore increases the diversity. And so what is this process called when bacteria take in foreign DNA and express it? Roman? It is not endocytosis. Starts with a T. Transformation. That's why this lab is a transformation lab. So we would say that this bacteria has been transformed after it takes in the plasmid. So imagine this, like if you have so you have all these plasmids, and they all have the two genes. So they have the human growth hormone gene, and they have the ampicillin resistant gene, and you're mixing these plasmids with a whole bunch of bacteria. I just drew one bacteria, let's draw a couple of other bacteria. So you have this clear solution with this in here. Can you be sure that every single bacteria in your solution took up the recombinant plasmid? No, all right? So you're just mixing it in there, giving it some time, and seeing what happens. So therefore, Can somebody tell me what's the purpose of using a plasmid that has the ampicillin resistant gene on it? Why do they choose? They specifically choose a plasmid that has that on there. What's the purpose of that, Matt?
If you cultural bacterial colonies in ampicillin, and that would kill off any bacteria that didn't take any plasmids that contain the ampicillin or common gene.
Yeah, absolutely. So when in our lab when we do this, when we have a solution with this bacteria and we give it time for it to take in the plasmid, you're going to grow them because you can't see the bacteria in a clear solution like this here. You can't see it, and so you know that there's some bacteria in there. You're going to take your Petri dish that has agar. And agar, the main source of food for the bacteria is LB. LB stands for Lurio broth. It's nutrients for the bacteria. And when I made the agar, I mixed this Lurio broth, this nutrients, with the agar, which solidifies and forms like a gel at the bottom of the Petri dish. So in this gel is this Lurio broth. So on LB, just LB agar, any type of bacteria can grow on that and eat off of it and so on. But what I did with some of the plates that we did is if you put ampicillin, so before it solidified, it was liquid, and so it was liquid to begin with, and when you pour it into the plates, as it cools down, because I heated it up to mix it and dissolve everything together, as it cools down, it solidifies, kind of like Jell-O does, and so, so while it was still liquid, I added ampicillin. So now, if this agar has ampicillin, which is an antibiotic, in with the food, and from this culture, all right, so you take this and you take some of the fluid and you spread it around on the agar and you let the bacteria divide. Which bacteria are the only ones that are going to grow on that? They're the ones that took in the recombinant gene, and so therefore it's a way to weed out bacteria, so to separate them out, okay? And that's what the ampicillin is used for. All right, so in our lab, as I said, we're gonna do the transformation part. So I ordered it's called the P-Glow plasmid. It's the plasmid that contains the green fluorescent protein gene. And so I already ordered this, all right, from the company, all right, that I ordered it from. It's called BioRad. So I have a recombinant plasmid. So we're not going to do this part. That's why I wanted to do that in the activity on Friday to kind of get it. That's what literally the company would have done, is to do that to make the recombinant plasmid. We're going to do the transformation part, where we're going to expose E. coli bacteria. We're going to use bacteria that is not harmful, and so it's from a company that's, you know, you're supposed to use it with students. Although we're not going to open the Petri dish and like touch the bacteria, okay? But it's not a harmful bacteria. And so, and we're going to do this transformation process and try and get bacteria to take in the recombinant plasmid. So if you look at your procedure sheet, I just want to talk a minute about what the company did before we got this plasmid. And you have a picture sheet that says the arabinose operand. What's an operand? What's an operand, Abby?
The clusters of genes controlled by a single promoter.
By a single what? Promoter. That's right. A cluster of genes controlled by a single promoter. We in this class have talked about the lac operand and the trip operand. Operands are only found in what kind of organisms? Bacteria, prokaryotic cells. And so the plasmid that this company chose to use has the arabinose operand as part of it. And so, there we go. So, to understand this and what the company did, let's look at this picture here. I'll enlarge this a little bit. Okay, so here's your operand. It has the 3 genes. The 3 genes code for making enzymes to break down the sugar. How do I know it's sugar? Enzyme-ose, right? So arabinose is a type of sugar. This is the DNA from the plasmid. So there are 3 genes. They call it ARA, stands for arabinose, and then we have VAD. This is what the plasmid looks like. So this is part of the plasmid. So if this is the plasmid here, when they isolate it from bacteria, it has the P stands for the promoter region, and then we have the VAD genes. That one promoter controls all of those 3 genes. And then the plasmid that they're using also contains the ampicillin-resistant gene. So this is what the plasmid looks like when the company isolates it from the bacteria that this plasmid is naturally part of. Okay, so just like we talked about before, with arabinose, if you're going to make enzymes to break down arabinose, it's only beneficial for the bacteria to make these enzymes when what is present? Arabinose. So here's the sugar arabinose. Now this ARA-C, this is blocking the promoter region. So right now, if there's no arabinose in the environment, in my loop here, I'm pointing with here is RNA polymerase. RNA polymerase cannot bind to the promoter because this ARA-C is blocking it. So therefore these would be turned off and you wouldn't make the enzymes to digest arabinose. If arabinose is present, look at what happens. The shape of the ARA-C changes. That shape change exposes the promoter region which means that now RNA polymerase can combine and transcribe the gene and make the mRNA, and therefore that can make the enzymes to digest arabinose. This is what's naturally found in nature and what happens in nature with bacteria that have this particular plasmid. So what the company I ordered from did is they took this plasmid from nature, they isolated it from bacteria, and they did 2 things to it. We look at here, we look at what it looks like. So this expression of the green fluorescent protein, this is after the company has changed this plasmid. Notice here that here's your ARA-C, still looks the same, it's still blocking the promoter region, but the 3 genes are out. So what they did, that's what these lines here, these 3 genes, they cut them out. So they cut the VAD out and replaced it with the green fluorescent protein gene. So now, this is what the plasmid looks like. The plasmid now has the same promoter, and it has the GFP gene instead of the VAD. So this promoter controls this instead of the VAD, all right? And it still has the ampicillin, they didn't touch that. So now, the GFP stands for green fluorescent protein. Transcription can only happen when this gene is turned on. This gene is only turned on when what is present? Arabinose, right? And so therefore, that's what we have here. Arabinose is present, changes the shape, and so on. And so this right here is the plasmid that I ordered. So it's already recombinant because it already has the jellyfish gene in, okay? So that's that. So what we want to do is put this plasmid into our normal boring E. coli and make it cool so that it glows. All right, so that's the thing that we're gonna do. So the next little part here, that's the background information. I want to make sure everybody understands. The next part here, I'm just gonna kind of go through. If you look in your packet, you have this picture. I love this. This company does a nice job with what to do and putting pictures as well with it. This is a little quick guide, but I'm not going to read through it all with you. You need to be prepared tomorrow, but I do want to make sure that you know where the materials are, what they look like, and so on, because like I said, you kind of need to know to get it done, so I don't want you going, what's this, you know, and so on, so you know exactly what you're supposed to be doing. So you're going to get a bin in your group, a green bin with your stuff that you need for the lab. So the first thing you're going to get is you'll have 2 micro test tubes. They look like this micro test tube, and they'll be in this foam container here that looks like this. It has 6 different slots in here. You'll have 2 that are open that are not labeled. You'll also have 2 more that have liquid in it. We'll talk about that more later. So the 2 that are unopened, you'll also have a Sharpie in here. So you're going to label 1 positive P glow and 1 negative P glow. So there's nothing in there. Positive P glow, positive means that's going gonna contain bacteria that is exposed to the recombinant plasmid. The pico means that that's the recombinant plasmid. So that's what I wrote up here. And then the negative test tube, you're gonna put bacteria, but you're not gonna expose it to the recombinant plasmid. So that's what the negative means inside, okay? On there, so you'll label those. Then it says you're gonna add this transformation solution, and it looks like you're gonna have a big old vial of it. You're not. That's one of the things you're gonna have. You're gonna have a little micro test tube. It's gonna be closed. It's gonna have liquid in it, and this is why I wrote right here. It's gonna be labeled trans. So I'm gonna write trans on here. So that's your transformation solution. So every group will have their own transformation solution, and you're gonna use sterile plastic disposable pipettes. They're sterile. They will not have been opened. You need to open them out of the container. Be very careful when you're reading the steps, that whenever it tells you to use a sterile one, use a sterile one. You should not be repeating this. So you don't want cross-contamination. So just take special care in reading the instructions there. So that's that. One other thing I want to point out is the readings on here, because they asked you to do 250 microliters. 250 microliters is 1/4 of a milliliter. So on the base of your disposable pipette, you will have 4 lines. The top one here near the bulb is 1 milliliter, and then the 3/4 half. And so there's your 250 microliter. And also notice here has it kind of indents going down, so the tip is pointed. So you want to count up there too and make sure that you're using the right line. And just a quick reminder about how to use these to get the correct amount in it. So before you put it into your little micro test tube, you're going to squeeze the ball, and then you dip it the end down into your solution and slowly let go with your fingers until the liquid will go up until it reaches the 250 milliliter mark, and then you take it out. Then you can let it go, or you can just squeeze it into the thing that you need to. So that's how you do that to get the right amount, all right? If you have any questions on that, let me know, because you want the right amount, and if you use too much, you'll run out of solution. All right, you'll have a styrofoam cup here for putting it on ice. It looks like a little tray here. You're going to have a styrofoam cup. I'll have a thing of ice in the sink here that you'll have to go and just scoop out some ice and put it in there. Notice that they made the thickness of the foam so that the bottoms of the test tube stick out. That's where your liquid is, so that it can stick in the ice. You can also take it out. They'll be closed. You can take it out and bury it in the ice too. That might be even a better thing. So then you're going to get your bacteria. Every group is going to start out with what we call a starter plate. I'm growing bacteria right now that I'll show you before you leave today. And each group will get their own little plate of bacteria. You're going to use a loop. They look like this. You have a whole package of these. And this is another one you want to make careful note when it says to use a new loop, okay? So, a new sterile loop. So, this is the loop. It looks like a very long, small bubble blower, all right? With the loop at the end. That's what it looks like, okay? And so what you're going to do is you're going to take your Petri dish, and the bacteria is growing on it. Remember, the agar is soft. So you just take this, and you can just gently scrape the top. Just barely touch it. You scrape the top and get some bacteria off. It'll look like a white slime on the tip of your loop. Okay, that's what the bacteria looks like. Be careful not to use too much pressure, because you'll gouge the agar. Okay, so just gently scrape it from the top. The bacteria comes off very nicely. And then you're going to put it in both of these, the positive and the negative, but let me just explain here. This is when you put it into the solution, you want to do like this, and keep doing this until when you come out, you don't see any of that floppy bacteria on it. So you want all the bacteria to come out into the solution. Okay? Number 5, the plasma DNA. I'm just going to have a classroom vial of the plasma DNA. That's going to be up here. So this is a solution that contains all these recombinant plasmids. It's going to be in a clear little, very small little glass bottle up here with a UV lamp is up here. So it's going to tell you, ask you if the plasmids themselves glow. So the glowing, the bacteria, in order to see the fluorescence, you need to expose it to UV light. So what's going to happen is that this vial, you can just take the UV light and shine on it and see if the liquid, the plasmids are glowing or not. So that's what that is. When you come up here though, the purpose of this is you're gonna put the plasmid in the you're gonna mix the plasmid with just the positive P glow bacteria. So notice here, the negative P glow, you're not gonna put any plasmid, so we're just with this. Two people need to come up here to do this. So one person holds the little vial that has the bacteria. The other person has to take the vial in a new sterile loop and stick it into the solution. You want to make sure that you see a film, so kind of hold it up to the light so you can see the reflection, so that you can see that there's a film over it, like you're blowing bubbles. And it needs to go right into the bacteria, that test tube, and then swirl it around to get all the plasmid out. And so therefore you'll be exposing that bacteria to the recombinant plasmid. That seems really easy. This is usually the part that most people have the most trouble with. All right? Because what you have to do, you have to make sure that it's the film thing. You have to make sure that you get a film and that film doesn't pop before you put it into the test tube. If it pops, then you have to do it again, all right? And so some people get it the first try, but it's very typical. I have students here like 20 times trying to get the film. So if that's you, don't feel so bad because it happens every year, okay? So it just may take a little bit of time to get that film, all right? All right, so then we have some downtime because you incubated on ice for 10 minutes. During that downtime, also in your bin, you're going to have these four Petri dishes, and I will have labeled what's in the agar. So you have these two right here have LB and ampicillin in the agar. This one right here has LB, ampicillin, and the sugar arabinose. And this one over here has just LB. So I already have that labeled. What you need to do when it says label your 4 agar plates, you need to label what's going to go in it, positive P glow bacteria or negative P glow. So that's what you are labeling, is the positive or negative P glow. And when you label it, you'll see when you get them that I labeled along the side. So when you look at it here, if this is the Petri dishes at the bottom, like I will have labeled like this LB amp, so you know that ampicillin and LB are in there. The reason why I put it along the side is because when you look at the bacteria, you're never going to take the top off. And so therefore we're just looking at the bacteria through the Petri dish, and you don't want that writing to be in the middle. So when you write positive P glow and negative P glow, you're also writing it on the side, okay? And we'll put names on it later, so you don't have to put any names on this, so you're just putting what's in there, all right, along the side. Okay, water bath. I have a automated water bath that's going to be on that pod over there by the sinks. I can set it for 42 degrees Celsius, and so what you need to do is, for this, you need, when you get to that end of that 10 minutes, the downtime, somebody with a stopwatch, one person with a stopwatch, one person with the flotation device. This is why they made these containers out of foam, because it floats on water. And so what happens is after the 10 minutes, one person with the stopwatch, one person with the thing, puts it in, starts at exactly 50 seconds, then you take it out and put it back on the ice. So it stays on the ice until you put it in, and then immediately goes on the ice. This is probably the most important step in getting the recombinant plasmid in. It helps for the uptake of it from the bacteria. All right, so that's important to do that. And make it go right from ice to water to ice. All right. The next step, it says here, you're going to add LB broth up 250 microliters. Again, it looks like you're going to get a big old vial, but you're going to get, I'll have a 4th little micro test tube. It's going to be labeled LB. So you're going to have one of these in your thing that's labeled LB, and that's where you get it. Okay? And then notice it's the same spot, 250. All right. So after you do that, you let it set for 10 minutes at room temperature, so you have some more downtime. And after that 10 minutes, you tap it. It says tap the closed tubes with your finger to mix. Let me explain what that looks like. All right, that's like this. All right, so what you're doing is mixing the solution down below. All right, so that you're doing that. All right, when you're ready to plate it, you take your 4 plates, go like this on your table to get all the liquid to the bottom of it, because when you tap it, it may splash a little bit along the side, so you can tap on the table to get it all back down to the bottom. And you're putting, as the thing says here, what bacteria and what plate. You're using 100 microliters. This is below, right below the first kind of little indentation below that 250. And so you're going to have this clear solution on here. You're going to use a new pipette for each one, so you have your clear solution. And then you get to spread it around. So that's using a new loop. So then here, you'll just spread it around, and then notice the lines here, then go this way, this way, this way. Spread it around as much as possible. When you're done with that one, get rid of that loop, and then you do it with this loop and another loop and so on and so forth. And so then you have your bacteria all ready to grow. So you're gonna pile them on top of one another, all the Petri dishes. The Petri dishes, the agar goes on top. So the bacteria, thinking of this, the agar's on top, so the bacteria are hanging out upside down, okay? That's because when you put them in the incubator, sometimes the agar releases some water and it condenses along the top. So if you put it the other way around, what happens is the evaporation causes, then it condenses on the top and drips on your bacteria. So you want them upside down. We're gonna masking tape them, so I'll have some masking tape available. You're gonna tape them all together, and that's where you're gonna put your names for your groups, so that you can then put them in the incubator in the back, and they'll sit for 24 hours till the next day, and hopefully they'll have some bacteria that are transformed, all right? And so that's that. So I just wanted to go through that, make sure you knew what everything was, where everything was gonna be at. I will have a bag hanging off from the overhead projector that all the contaminated stuff can go into, so like all your loops and things like that as you use them. All right. So the next thing I'd like to do then is to have us get into lab groups, and I have enough for 8 groups. So groups cannot be more than 4. Okay. So I didn't count here for the number of people here. Let me see. All right, there's plenty for 8 groups. So what I'll have, we can get into groups of 3 or no more than 4. So 3 or 4. And so I'm gonna let you guys pick your groups. So before we're gonna do some hypothesizing in just a second, but right now I want you to just get into groups. So I know who's in what group, and then I'll talk about the hypothesis, okay? So go ahead and organize yourself. So we need, all right. So you guys, we're going to hypothesize together so you can bring it forward. Okay. And you got what's
She goes,
Alright, and you guys Alright, I don't have enough. Let me count.
Friday? Oh, that's another tough.
You guys, you 4?
Yeah.
4. You 4? 5. 3. Yeah, 6. I have 2 people who are absent. I have Savannah and I have Dallas. So I may have them join a group of 3. Okay. Alright. So you guys are good. Okay. Alright. So look on the sheet that you answered the 4 questions on. Guys, look at the sheet that you answered the 4 questions on. And look at the second page, like the part that says, has a chart that looks like this, pre-lab prediction. So what we're going to do is we're going to predict what's going on and what's going to happen in the lab. So notice that the top, you have rows that are labeled, but they're not labeled at the top. And so, let me put this back up here. So what we're gonna do is label it based in the same order that we have the four dishes. So the top left one, we're gonna leave blank, and so we're gonna label above these 4 columns. So the first column is gonna be positive P glow and LB/amp. The second column is for the second Petri dish, so positive P glow, LB, amp, and era. The third column is for the negative P glow with LB amp. And the fourth column is negative P glow with just LB. Remember, as a reminder, the positive and negative P glow are telling you about the bacteria, whether or not they've been exposed to the recombinant plasmid. The LB and anything behind it is telling you what's inside the agar that it's going to grow on. All right, so that's the information that you have. So what I want you to do is think about each one of these 4 plates. The first row says purpose of the plate. So what I'd like you to do is think about, why did the manufacturer and why does the lab even have this as a plate? Like, what's the purpose or role of having and plating bacteria on that plate? All right, so that's what I want you to think of first. The second thing, the second row, says growing. So growth. Do you expect bacteria to grow on there? If you put no, if you don't expect it, then you're done with that box. If you think that bacteria is going to grow, I want you to tell me what kind of bacteria. Is it going to be transformed bacteria? What does that mean? Transformed bacteria, what do you think? They took in the plasmid. Do you think it's non-transformed bacteria, bacteria that didn't take in the plasmid? Or do you think both of them are going to grow on there? So what kind of bacteria grows on there? And the last thing is whether or not you think it's going to glow or not. And the glowing is under fluorescent light. Okay, so I'm going to give you time. You can work with your lab groups. What I'd like you to do is discuss it. Some people may have a hypothesis right away. Other people may have to think about it more. So if you're a person that feels like you get it a lot and you can explain it, make sure that other people, everybody in your group understands what's going on. If you're having trouble, don't just copy a hypothesis from your lab group members just because. So have them explain and so on. Okay?
No.